Hello everyone. So the next topic which we will be studying is about the mechanism of the filtrate. So what we will be studying in this is about the tubular reabsorption. So what is it? We will be studying this in detail. That tubular reabsorption is the process by which the solutes and water they are removed from the tubular fluid and just being transported into the blood. Now there comes a term which is called as the filtrate. So we will be discussing the mechanism of this filtrate in detail. So what is filtrate? Filtrate is a fluid which has been filtered from the blood and they passes through the nephron, and much of the filtrate and its content they are reabsorbed into the body. So there is a process or mechanism of reabsorption which we will be studying. So reabsorption, it is a finely tuned process that is altered to maintain the hemostasis of the blood volume, blood pressure, plasma osmolality and blood pH. So this is the main function of the process of reabsorption, that what it does is it helps to maintain the hemostasis of blood volume, blood pressure and plasma osmolality or the blood pH. Now there is a reabsorbed fluid, ions and molecules that are being returned to the bloodstream through a peritubular capillaries, and they are not excreted as the urines. So what is the mechanism of the reabsorption? The mechanism of the reabsorption is that the reabsorption is the process in the nephron that can be either active or passive, and specific permeability of the each part of the neuron they varies considerably in the amount and the type of substance which is being reabsorbed. Now the mechanism of reabsorption into the peritubular capillary includes the three mechanisms. And what are these three mechanisms? First is the passive diffusion. So the first one is the passive diffusion. So we have already discussed that in this sentence only. So what is passive diffusion? It is the passing through the plasma membrane of the kidney epithelial cells by the concentration gradients. So this is called the passive diffusion. And if I talk about the active process here or active transport, it is the membrane bound ATPase pumps with carrier proteins that carry substances across the plasma membrane of the kidney epithelial cells by consuming the ATP. So this is known as the active transport. And there is a term which is also called as co-transport. So what is that? So in the process of co-transport, there is a reabsorption of water that takes place. So water, they follow other molecules that are being actively transported, particularly glucose and sodium ions in the nephron. So the three mechanisms or the three processes which we have studied, they involve the substance passing through the luminal barrier and the basolateral membrane, that is the two plasma membrane of the kidney epithelial cells, into the peritubular capillaries on the other side. So some substances can also pass through tiny spaces in between the renal epithelial cells, which is called as the tight junctions. Now what we will be studying is about the mechanism, that how this mechanism takes place. So I have described about the three mechanisms here, that is the passive diffusion, active transport and the co-transport. Now what is this figure talking about? So this is a diagram which is showing the basic physiologic mechanism of the kidney and the three steps that are involved in the urine formation, namely filtration, reabsorption, secretion and the excretion process. So there is a glomerular capillaries which is being depicted here, Bowman's capsule, and the four steps which we are discussing here. So as you can see in the step 1, there is a step 1 which takes place, and there the 2 and 3 and 4 are being displayed. So here what is happening is, first is about the filtration step. So there is a filtration which takes place in the first step. Second step is the reabsorption step, which we have discussed here, and there are three mechanisms that are involved in the reabsorption step. And what are the three mechanisms? That is the active transport, passive diffusion, and the co-transport. The third step is about the secretion, and the fourth step is about the excretion, which is in the form of the urine. So in the fourth step, there will be excretion of the urine that takes place. Now, about the osmolarity changes. So we are talking about the main motive of our this is the mechanism of the filtrate. So as the filtrate passes through the nephron, its osmolarity changes as ions and water are reabsorbed. So there will be a change in the ion and water because they are constantly being reabsorbed. So the filtrate enter into the proximal con convoluted tubule, which is about 300 MOSM per liter, which is the same osmolarity as normal plasma osmolarity. So it will be the same as the normal plasma osmolarity. And if I talk about the PCT, that is the proximal convoluted tubules, here all the glucose in the filtrate is being reabsorbed. So what is being reabsorbed here? It is the glucose which is being reabsorbed along with an equal concentration of ions and water, so that the filtrate is still 300 MOSM per liter as it leaves the tubule. So the filtrate osmolarity drops about 1200 MOSML per liter as water leaves through the descending loop of the Henle, which is impermeable to ions. Now, so first is the PCT, that is the proximal convoluted tubule. The first step is that. Then comes the loop of the Henle, and after that comes the distal convoluted tubule. So in the loop of Henle, which is permeable to ions but not water, so the osmolarity here will falls to about 100 to 200, the same the SI unit which we have been using this. And finally, after the Henle tube, loop of Henle, then comes the distal convoluted tubule. So, and the collecting duct, where a variable amount of ions and water are reabsorbed depending on the hormonal stimulus. So the final osmolarity of the urine is therefore dependent on whether or not the final collecting tubules and ducts are permeable to water or not, which is being regulated by the process of the hemostasis. So this was all about the mechanism of the filtrate, and these are few references.